All right. We are starting to record here. Today what we're going to do is start with regulation of ion and water balance. Thank you so much for your patience, everyone. Crazy morning. All right, so let's get right to it. Today what we're going to be talking about is ion and water, okay? So we're going to be actually talking first about how the loop of Henle establishes that profound osmotic gradient within the kidney, within the medullary region. So, how many of you caught that? How many of you understood what I said just now? Okay, so that's good. That's very telling. Okay, so here's what I want you to remember from last time. Does everyone remember that there is a profound osmotic gradient within the medullary region? Okay, so what I mean by the gradient is right here in the medullary region, very close to the cortex, it's only about 300 milliosmoles. So it's not that concentrated, right? But if you actually get down here in the lower end of the collecting duct, right before you get to the renal pelvis, this is very concentrated. The solute concentration in this area can be as high as 1,200 milliosmoles, 4 times what it is up here in the cortex region. Okay, so this osmotic gradient is what is allowing you to concentrate urine. Without that gradient, you have no capability of concentrating urine, okay? So that's what we're talking about right now. We're talking about how this loop of Henle, right? This is part of the nephron. Let me just go through the structure one more time. Glomerulus, proximal tubule is right here. This is where all the reabsorption occurs. Then the loop of Henle. These juxtamedullary nephrons, the juxtamedullary nephrons actually have these long loops of Henle that extend deep into the medullary region where it's very concentrated and then come back up into the cortex region, okay? This is a different type of nephron right here. This is also a nephron, but it's called the cortical nephron. And it really only remains in the cortex region. That's why it's called a cortical nephron. These types of nephron do not participate in establishing that profound osmotic gradient in the medullary region. Only the juxtamedullary nephron have that capability. All right? So these loops of Henle, the loops of Henle of these juxtamedullary nephron are responsible for establishing that profound osmotic gradient. And that's what we're going to be concentrating on today. That's what my first sentence was. I just want to make sure everyone is on the same page. Okay? So also I want you to notice the vasa recta here. Okay, we'll be talking about the vasa recta in just a second. This is the blood supply, and it's named the vasa recta. It has a special name because it only surrounds the loop of Henle of the juxtamedullary nephron. And it has a special name because it helps to maintain that osmotic gradient that's established by the loop of Henle. And we'll talk more about that as well. Now, the vasa recta really isn't a nice U shape like this. I mean, it's nice to diagram it this way, but it actually looks more like this. It completely surrounds the loop of Henle. Okay, so just so you know that, I want to make sure. And don't forget when you have reabsorption going on, reabsorption pulls molecules and solute and fluids out of the tubular lumen. It has to cross the epithelial cells and the interstitial fluid and then be delivered into the capillaries, right, into the vasa recta. So there's a number of different cell types when that solute and fluid is being reabsorbed. We just want to make sure everyone is on the same page with that as well. All right, so let's take this particular diagram that we've seen in the past, right? This is our tubular tour. And what I want you to focus on is the loop of Henle. I'm going to give you a different diagram now to show you how exactly the osmotic gradient is actually established. So remember the thin loop here. If we remember what the thin loop of Henle does, is it is highly permeable to water and solutes. So usually water flows out as it encounters a more hyperosmotic environment, right? Just from osmotic forces, osmotic driving forces, as the fluid in the thin loop comes across, actually encounters a more hyperosmotic environment, could be as much as 1200 milliosmoles, that's going to tend to pull water out. Solutes are going to equilibrate, they're going to move in and out until the solute concentration within the loop is also about the same as it is outside. All right, the thick ascending limb, however, pulls salts out, but remember, it has very low water permeability, so the water is not allowed to follow. This is called the diluting segment because it pulls salt out, but really the water remains inside the tube. And when you get to the top of the distal tubule here, very close to the glomerulus, you can actually see it's only about 150 milliosmoles. It's very dilute. All right, so how does that look, right? This is what's happening. You can see as you go down the thin descending loop, water is being pulled out, salts are moving in, and then as that fluid rounds the bend and starts to go into the thick ascending limb, most of the salt is actually pulled out in the beginning of the thick ascending limb than it is at the top. So you can already see that the thick ascending limb is starting to establish that osmotic gradient within this particular medullary region, right? And it's actually due to active transport mechanisms because the pump is involved. The pump on the basolateral membrane is actually pumping sodium out. Chloride is following via the paracellular pathway. Okay, and so salts are being pulled out of the thick ascending limb and actually establishing this profound osmotic gradient. Okay, so let's take a closer look just to help solidify this concept. This is a different diagram from your textbook. It actually is showing you, here's the proximal tubule, the loop of Henle, and the distal tubule. Okay, so if the proximal tubule has, right, when you filter fluid, it's about the same as plasma, about 300 milliosmoles. But then as it rounds the bend here, the red arrow is signifying active transport mechanisms. It's pulling salt out, okay, and then you can actually see that helps to increase the driving force for water to be pulled out. So remember that salt is actually staying in the interstitial fluid as well. And when we elongate and look at these forces, let's elongate that tube, you can actually see as you go down the descending limb, water continues to be pulled out, concentrating the fluid inside the tube. And it's all due to these active transport mechanisms that are pulling salt out but not allowing the water to follow. Now you can see how this profound interstitial osmolarity is established by the loop of Henle. And that's again why some of those desert animals like the kangaroo rat, like we talked about, have one of the longest loops of Henle, right? Because it has to establish this profound osmotic gradient in order to concentrate and conserve water. All right? So You've learned some of these concepts before. I'm just layering on, so that you can actually see why the loop of Henle is so important in actually establishing this interstitial osmolarity, this osmolarity gradient. All right, so here's the key to understanding this as well. Now taking a look at the entire nephron, right? We've got the proximal tubule, the loop of Henle that's establishing the osmotic gradient. Here's the distal tubule and the collecting duct. You can actually see even in the collecting duct, this whole area down here is 1,200 to 1,400 milliosmoles, very concentrated. All right? So we're not just talking about what's happening in the local environment within the tube and just right outside the tube. We're actually talking about this entire layer. Even it has effects on the collecting duct. So all of this interstitial fluid across this entire region is now very, very concentrated. So it actually has effects on the collecting duct as well. It tends to, if water permeability is there, right, then it will tend to pull water out, reabsorbing water, to form a very concentrated urine. Okay? All right, so this also shows you, this is a nice diagram showing you that this entire region is very concentrated with solute. So here's the nice osmotic gradient, about 300 milliosmoles towards the cortex region. But as you dive deep into the medullary region, this entire region gets more and more concentrated. I'm going to say it, the technical term, it's more hyperosmotic in this region. So the loop of Henle and the collecting duct, this actually acts as a countercurrent multiplier. Okay, so that's a new term. Because it uses active transport mechanisms and they go in different directions, right? This thick ascending limb is going in the opposite direction as the thin descending limb and the collecting duct. These are called countercurrent multipliers. And they create, that loop of Henle creates that osmotic gradient that facilitates transport processes. Okay, so before we get into that, yeah, question? Yeah. Yep. Okay, so countercurrent means that the direction of flow is actually going in opposite directions. Okay, so when we're talking about the thin descending limb, you can see it's actually going towards the inner medullary region. And then the thick ascending limb is going in the opposite direction. So that's what's called a countercurrent. And because of that countercurrent and the active transport mechanisms, the pump is really involved in this. Let's go back to this. You can actually see the active transport mechanisms pull more salt out in the beginning of the thick ascending limb. So there's way more salt in this area than there is here in this area, and then there is here. Okay, so it's establishing that osmotic gradient because of the countercurrent. And it's called a multiplier because of the active transport properties. It requires ATP. The hydrolysis of ATP provides energy for the movement of that salt out. And that, again, is what's establishing that osmotic gradient. So countercurrent is just referring to the opposite flow of those tubes. Okay, so let's take a look at the vasa recta now. The vasa recta. Okay, remember that is the blood supply. This always is a little confusing. I know I see this a lot when students are first learning this concept. The vasa recta is the blood supply that surrounds the nephron. So just to give you an orientation, this yellow tubes is the loop of Henle, and the red and blue, this is the vasa recta. Okay, so let's concentrate on the vasa recta for a minute. Just so you know, the take-home message here is the vasa recta is organized in such a way that it maintains that osmotic gradient that's established by the loop of Henle. So because of this U shape, it's also a countercurrent, right? Blood is flowing down towards the intermedullary region and then back up towards the cortex. Because of that U shape, it helps to maintain the osmotic gradient that's established by the loop of Henle. All right, so going forward, here you see on the left-hand side the nephron, and here's the loop of Henle, the thin descending loop and the thick ascending loop. Now, this diagram is not the way the vasa recta is organized. This is not the way it's organized. If it was linear like this, the blood would actually take up salt, right? It would take up more and more salt as it entered into the intermedullary region. And if it was linear like this, it would just wash the salt away, right? It would just erode that osmotic gradient that was established by the loop of Henle. Water would be pulled out, salts would actually enter into the tube and then just be washed away. Okay, but that's not how it's organized. It's actually organized in this U shape. So it takes up salt, right? Water is actually forced out of the tube as it encounters a more hyperosmotic environment. But then it takes a turn. Okay, it actually takes a turn and it goes up towards the cortex region again. Okay, up towards the cortex region. And as it ascends towards the cortex region, water actually enters into the tube and salts actually are delivered out. So when you get to the top of the vasa recta, again towards the cortex region, you really haven't lost that much salt, right? You go from about 310 milliosmoles to about 320 milliosmoles. Perfect, elegant, right? Exquisite. It helps to maintain that osmotic gradient that's actually established by the loop of Henle. This is a little more complicated slide. Okay, this slide is actually just kind of giving you some numbers as to the osmolarity within the tube, within the vasa recta. As it descends, remember water is in the dashed lines. Water is being pulled out while salts are entering in. Salts are the solid line till it gets very concentrated. So I want you to think about that for a minute. Think about a red blood cell, erythrocyte. Remember, erythrocytes are red blood cells? Think about a red blood cell flowing down towards the intermedullary region, encountering a more and more hyperosmotic environment. That red blood cell is going to be pretty shrunken up, right? It's going to be very shrunken, crenated. All right? That's some profound cell volume regulation, right? Think about that. Cell volume regulation in a red blood cell. And then as it rounds the bend, right, it goes back up towards the cortex region and salts are pulled out, water enters in because, now these are just passive forces. Passive forces. This is called a countercurrent exchange. Countercurrent exchanger. All right, so the take-home message, these two, let me just sum it up again. These are not easy concepts. The loop of Henle is called a countercurrent multiplier because of those active transport properties that are establishing the osmotic gradient. But the vasa recta is known as a countercurrent exchanger because it only relies on passive forces. Passive diffusion, passive osmotic gradients, right? Osmotic driving forces that are moving solutes in and out and water in and out. Okay, so countercurrent exchanger is a term describing the vasorecta, only because this is completely passive transport. So again, what is the significance of the countercurrent exchange system? Why is the vasorecta important, and what is the significance? So we actually had a discussion about this already. Why do we have the vasorecta? Why do we have this very specialized blood transport system that surrounds the loop of Henle? Why is that necessary? Yeah. To maintain the osmotic gradient, absolutely. Number one. Perfect. For reabsorption and secretion, absolutely. Right? When you reabsorb or secrete molecules, you have to have the blood supply right next to it. One more thing. One more thing. What do you think? What about those epithelial cells of the loop of Henle? They need oxygen and nutrients, right? You have to have the blood supply right there to provide oxygen and nutrients to those cells of the loop of Henle. Okay, so that's also very important. Okay, so think about this. What is the significance of the countercurrent exchanger? Here's another analogy that you can think about. This is heat transfer in the extremities, all right? So when you think about the blood that has to perfuse the cells of your fingertips, right? Usually in the arteries, the blood is very warm as it enters into your arm. But especially cold days like this, right, in the wintertime, as it travels towards your fingertips, it cools down. It cools down. But then as it travels back up towards your body, the arteries and veins are right next to each other, right? So the cold blood from the veins is warmed up by the warm blood in the arteries. Countercurrent, they're close to each other, and the blood warms back up in the veins as it travels next to the arteries back towards your body. That actually helps to keep your core heated properly, right? It helps to keep your core heated properly. That's another example of a countercurrent exchange. Okay, countercurrent exchanger. All right, so these are not easy concepts, right? Now we're kind of getting into the nitty-gritty of the nephron. All right, so let's keep going a little bit here. I also want to show you, when we were talking about excretion, right? When we were talking about excretion, in fact, I didn't finish excretion, so we'll revisit that in the next lectures or so. Let's talk about urea, though. I did mention urea on Monday. And I actually mentioned that it was an important osmolyte within the kidney. You can actually see that urea is recycled in the kidney, right? The urea that's filtered into the glomerulus is reabsorbed, just like a lot of other molecules. About 50% is reabsorbed in the proximal tubule. It's very concentrated as it enters into the thick ascending limb. You can actually see about 50% of that is also reabsorbed in the collecting duct and enters into the loop of Henle. So when it enters into the thick ascending limb, it's about the same amount that's filtered at the glomerulus. And then on the back end, if vasopressin is on board, you actually get a profound reabsorption of urea, again to help maintain this osmotic gradient. If you're reabsorbing a lot of water, you don't want it to dilute out your osmotic gradient. So urea is also reabsorbed to help maintain that profound osmotic gradient. And we'll talk more about this when we get to vasopressin. Okay, so just know from this slide that urea is also a very important osmolyte that helps to maintain the osmotic gradient. All right. So we're going to talk about, when we talk about extrinsic mechanisms, we're actually going to talk about vasopressin and aquaporin channels. Before we get to that, though, I want to make sure that you understand these terms. When we get to intrinsic and extrinsic mechanisms, I want you to know exactly what a diuretic is. We talked about Lasix last time, a powerful diuretic. I want to formally make sure that you know what a diuretic is. A diuretic actually stimulates the excretion of water. So what that means is it allows your body, when you take a diuretic, to form a very dilute urine that has a very large volume. So when you want to get rid of a lot of water in your body, take a diuretic. Like I said before, Lasix, the term pee like a racehorse, sure that it was a racehorse that was on Lasix. Okay? It just produces a very dilute, clear, it's not very concentrated, dilute, clear, large volume of urine. An antidiuretic is the opposite. An antidiuretic allows you to concentrate urine. It reduces the excretion of water by allowing your body to reabsorb water and deliver it back into your circulatory system, interstitial fluid and circulatory system. So an antidiuretic reduces excretion of water and allows you to concentrate urine. So what that means is usually the urine that's produced is very low volume. It's has a lot of color to it, very concentrated. So if you took the specific gravity, which was really measuring the solute concentration, it'd be very high, very concentrated. All right, so we're also going to be talking about steroid hormones going forward and peptide hormones. Steroid hormones, and we'll learn this in endocrinology too. Steroid hormones, they don't need receptors on the plasma membrane. These are lipophilic or hydrophobic molecules, right? I'm saying the same thing. They prefer to be in a lipid environment. Lipophilic or hydrophobic. And they can easily cross the plasma membrane, enter into the nucleus, where usually the steroid hormones have receptors. And a lot of times they initiate protein synthesis. And it's a very slow response because it takes about 24 hours for those proteins to be upregulated and expressed. So steroid hormones have a very slow response. It takes about 24 hours. Peptide hormones, we're going to be talking about vasopressin in a minute. Peptide hormones are derived from peptides, as the name suggests. They are hydrophilic, so they don't enter into the cells very easily. They usually have their receptors on the plasma membrane. And a lot of times they're like G protein coupled receptors, so the response is very rapid, right? Once it binds to its receptors within seconds, minutes, you can actually see the result. Peptide hormones. Vasopressin is actually called arginine vasopressin. Or if you read it in other journals, I'm going to write this all down, so don't worry if you don't absorb this right away. But arginine vasopressin is also called anti-diuretic hormone. It tells you its action. right? Antidiuretic hormone, or ADH, is exactly the same as arginine vasopressin. Exactly the same thing. All right, so I'll leave you with this. We're like I said, on Friday, remember, we don't have class. Unfortunately, I really want to keep going with this subject. Vasopressin is going to be one of the topics that when I talk about extrinsic mechanisms, just to give you a little bit of a taste. Here's what arginine vasopressin does. Let's just take a look at this slide. You've seen it before. Arginine vasopressin, when it's circulating in your body, inserts water channels in the distal tubule and collecting duct. This allows you to concentrate urine, right? Because if you have these water channels, you can pull water out of the collecting duct and reabsorb it to form a very concentrated urine, right? Does that make sense? Vasopressin is a hormone that's usually secreted when you're very dehydrated. When you're dehydrated, it inserts these water channels into the collecting duct so that you can reabsorb water. So you can pull water out of the collecting duct and form a very concentrated urine. So here's something interesting. I'm sure that only students that are older than 21 can understand this. All right? All right. Students younger than 21 will have no idea what I'm talking about. But let's say you go to a bar, you start to drink beer, right? You're drinking beer or alcohol, and about a half an hour later, what do you notice? That you're peeing constantly. The technical term is breaking the seal. Right? All right. So what is your body actually doing? Alcohol inhibits vasopressin. I'll say that again. Alcohol inhibits vasopressin. So now you don't have any capability of concentrating urine, and you pee constantly for a while, right? Which is why you're probably pretty dehydrated the next day. Have a headache, hangover, right? Yes, question. Oh, no. Okay. All right, no question there. All right, so you can impress your friends at the bar. When you drink alcohol, you're inhibiting vasopressin. Believe me, I've done it. All right, so that gives you a good idea of what alcohol does to your body. I think I only have a few more minutes, so what I'm going to do is revisit excretion. We'll finish that, and your video for Friday, just so you know, your video for Friday is going to include intrinsic and extrinsic mechanisms and basically a quiz associated with that. Pretty good? All right. Yes, question?
Is there like a certain time the quiz is due?
No, I think what I'll do is I'll have the quiz due Monday when I get back. Monday at 8:30. Okay, so you'll have the entire weekend to watch the video and then take the quiz. Sound good? It might be a situation where you have to watch the video before you take the quiz too, and it'll open up the quiz once you've watched both videos. All right, so let's go back to the intro section. I want to finish up with the bladder here. Here's where we left off. I just want to be able to go through this particular, there's only one more slide that we didn't get to last time. We're going to revisit right now the autonomic nervous system. This is the process. Oh, I had a really good question, right? I had a good question last time. I want to make sure that everyone realizes that secretion and excretion are different, okay? Secretion and excretion are different. So secretion is actually when you are taking molecules from the capillaries, right? Molecules and fluid, and delivering it into the nephron to be excreted. All right, so it's the process of transporting molecules from the capillaries into the nephron to be excreted. All right, so this is the process of secretion. Secretion. Excretion is when you are eliminating fluid. You're actually eliminating fluid from your body through the urethra. Okay, so that's excretion. We talked a little bit about these molecules, nitrogenous wastes. Okay, but this is the actual process of excretion. Here is a picture of your bladder. It's actually regulated by both the parasympathetic and the sympathetic nervous system and skeletal muscle, your motor, somatic motor neurons and muscle, skeletal muscle. So the smooth muscle that surrounds the bladder is actually called detrusor muscles, and they're regulated by the parasympathetic. So during filling, during passive filling, when the urine is entering from the ureters into the bladder, these muscles are inhibited. But during micturition, during the process of urination, you stimulate these muscles to contract. You're not doing this. This is actually involuntary. Remember, this is all through the parasympathetic, resting and digesting. Remember, this is called, the acronym was SLUD. Secretions, lacrimations, urination, and defecation. So this is part of the parasympathetic resting and digesting. It's inhibited during filling, but stimulated to contract during urination, micturition. Now, you also have these internal, they're inside the urethra, internal urethral sphincter muscles. These are also smooth muscles that are controlled by the sympathetic. So during filling, passive filling, they're stimulated to contract. But when you urinate, they're actually inhibited and they relax. You're not controlling this, right? These are just, during micturition, they're inhibited, they dilate to allow for flow. Nothing should happen until you actually control the external urethral sphincter. These are under skeletal muscle control. You're controlling this. You learned this when you were 2 years old, right? You learned how to control this. It's stimulated during filling, but you've learned to inhibit them to relax when you urinate. Okay, so that's under your control. This is voluntary. Internal urethral sphincter is under smooth muscle control. It's controlled by sympathetic neurons, but you control the external urethral sphincter, and you relax them during urination. Pretty good? Wow, what a morning. All right, so what we're going to do then on Friday, intrinsic and extrinsic mechanisms. It'll be 2 videos and about 50 minutes long, and then you'll have a quiz. Yes? Yes. No, we're going to put the problems off until Monday, and we're going to work on it as a class. Okay, so we'll be talking about a little bit of acid-base regulation and clearance equation on Monday. All right. Have a good weekend. I won't see you until Monday.